FAS FSL mediated cytolysis. FSL binds to the FAS recruiting pro-caspase-8 via the FADD2 from the disc, which activates the caspase-8. Caspase-8 triggers the apoptosis by cleaving the caspase-3 and BID. In type 1 cell thymocytes, caspase-8 cleaves caspase-3 directly, while in type 2 cell, that is the hepatocytes cells, it cleaves the BID to produce TBID, which induce the apoptosis by interacting with the BCL2 protein family and releasing cytochrome C from the mitochondria. Now cytochrome C activate the APF1, the protein present in the cytoplasm, forming the apoptosomes, where they form the apoptosomes when cells are in stress or damage the DNA, which now apoptosome activate the caspase-9 and subsequently caspase-3. Caspase-3 cleaves the ICAD, activating CAD to fragment DNA in the nucleus and hallmark of apoptosis.